Your Royal Highness, Princess Mahashak Visiridon, Your Royal Highness Princess Dina Miret, Excellencies, distinguished guests, ladies and gentlemen. I am deeply grateful and extremely honored to receive the Prince Mahidol Award in Medicine 2019. It's an extremely high recognition of my work, and although it is given to me, I accept this prize on behalf of all the colleagues in my laboratory who contributed over many years to all the work that finally has led to the discoveries that have just been mentioned. I'm pretty sure without all their contributions, I would not stand here. Now, one of the biggest privileges I enjoy as a scientist is to follow individual passion. In my case, it was biology, and more precisely, virology, and especially viruses infecting the human liver. This fascination already started at school when I met a teacher, nowadays you would call it a mentor, who raised my interest in botany and zoology. And after finishing high school, I began to study biology at Heidelberg University. And during this time, it was the early 1980s, I became very interested in plant sciences, but it turned out molecular plant studies were not possible in Heidelberg, and therefore I resorted to a topic of equal interest, which was infectious diseases. So fortunately, there was one laboratory in Heidelberg that was working on hepatitis B virus, headed by Professor Heinz Schaller. I found this topic most fascinating, and I got the opportunity to join his lab to do my PhD thesis that was dedicated to the molecular aspects of hepatitis B virus replication. During this time, I received an excellent training in molecular biology, and I learned how to engineer viral genomes, which was a skill that became very important later on in my career. In 1990, I moved to a big pharmaceutical company to establish a research program on an agent that was originally called the non-A, non-B hepatitis, and later on, after its discovery by Mike Houghton and his team in 1989, it was called the hepatitis C virus. This discovery by Mike and his team set the stage to establish diagnosis tests to screen blood and blood products, and it, of course, at the same time opened the arena for molecular studies of this virus. One of the first tasks was to delineate the genome organization of this virus and to identify the viral enzymes that could serve as targets for antiviral therapy. Initially, I focused on the viral protease, which is called the NS3 protease, and characterized the molecular determinants. Then I decided to go back to academia and to continue studies at the university in Mainz with my own research group focusing on another drug target, which was the NS5B polymerase. Now, these studies in vitro progressed pretty well, but we were all hitting a major roadblock. And this roadblock was the lack of assistance to culture hepatitis C virus in simply to propagate cells. Now, viruses are obligate intracellular parasites, and it's not possible to study them out of the context of a cell. And therefore, the testing of antiviral drugs was not possible without such an adequate cell culture system. For that reason, many laboratories in the world aimed at the establishment of an HCV cell culture system, and we were no exceptions to that. It turned out to be a long and windy road, and after more than 6 years of intense work with trial and error, we finally came up with a solution, and this was mainly driven by Volker Lohmann in my laboratory, who was able to produce a genetically engineered hepatitis C virus genome that was capable to replicate with a very high efficiency. Now, being a basic scientist, I have to say there are only a few eureka moments that you have in your scientific life, but I remember the day when Volker came out of the laboratory and showing evidence that indeed we had been successful in establishing such a culture system. And my response was, I just got goosebumps and didn't believe that we finally had solved the problem. Now, this achievement opened many doors for research and development, And within a short period of time, this system became a standard in research laboratories, in academia, and in industry. And in this way, antiviral drugs could be developed, the first one, or one of the first one being, for instance, inhibitors of the protease, like zimaprevir, and later on polymerase inhibitors like sofosbuvir. I personally think the biggest surprise in all these drug developments were compounds or drugs that are targeting yet another viral factor, which is called NS5A. And these antiviral drugs have turned out to be the most potent drugs that have ever been developed. And given that potency, they are now a real cornerstone of all antiviral therapy of chronic hepatitis C. Now, the development of these so-called DAAs was a true game changer in the clinic, because before the availability of DAAs, patients had to be treated with a combination therapy composed of interferon and ribavirin. Now, this therapy has numerous drawbacks. First of all, there was no guarantee for success. Around 40% of treated patients were able to eliminate the virus, 60% were unable to do so. The success rate was very much dependent on the so-called genotype. It required cost and labor-intensive diagnostic tests, and it had a lot of contraindications and side effects. And for that reason, patients had to be monitored very closely, and therapy was very limited by all these side effects, and many people simply had to stop antiviral treatment. And it's really difficult to imagine, because this therapy had to be given for a full year, that you can accept so many side effects without having a guarantee that later on the virus would have disappeared. Now, all these problems completely vanished with the development of DAAs. They achieve virus elimination in 95% of patients and even more in a routine clinic daily practice within a 12 weeks treatment. Therefore, for the first time, it became possible to cure a chronic viral infection. HCV targeting DAAs are also extremely well tolerated, have minimal contraindications, and can be given to patients even with advanced liver disease, liver cirrhosis, and even to patients with hepatocellular carcinoma. So for all these reasons, there is certainly medically no justified argument to use interferon-based therapies anymore. Moreover, the therapy of chronic hepatitis C changed from a complicated therapy with many side effects to a very simple therapeutic scheme. And it can now be provided by physicians outside specialized centers. This is most important for people living in more remote rural areas where medical care is much less available. For these reasons, DAAs and the therapy of chronic hepatitis C has become a unique opportunity to control or even eradicate a virus on a global scale. WHO has declared its aim to reduce the number of new HCV infections by the year 2030 by more than 90%. Now, unfortunately, thus far, only few countries are really on track to reach this goal, and many obstacles have to be overcome. Probably one of the most challenging problems is to make these drugs available to all countries and patients. In this regard, a major limiting factor has been and still is the price of these drugs. I remember the time, it was in 2014, when sofosbuvir was introduced in Germany for the price of 80,000 euros per treatment, which ignited extensive discussions about pricing of medications, our healthcare system, and how to tame extensive costs. Luckily, with the development of equipotent drugs by others, prices went down and have reached now around 30,000 euro in Germany per routine therapy. Nevertheless, for most countries, many of them having a high prevalence, such prices are unaffordable. As a result of the extensive discussions and negotiations at several levels, companies started to grant licenses to produce generic HCV drugs, for instance in India, which enabled the introduction of generic DAAs, for instance in Egypt and India, in the price of that range between 25 to 80 US dollar. So this is a great achievement, but there is still quite some unequal distribution between the countries, and in many countries, also including Thailand, currently the prices are still too high. Apart from the pricing, what is also required is a proper medical infrastructure to diagnose the infections. This is a very important task, since so far at most 80% of all chronic HCV infections have been diagnosed. Unfortunately, most infections are asymptomatic, and therefore people are simply not aware of their infection. Nevertheless, they have a high risk to develop end-stage liver disease, which includes cirrhosis and even liver cancer. Diagnosis and treatment, therefore, must be provided very early, which requires an adequate medical infrastructure, but also the financial resources to establish and sustain such programs. In that respect, the implementation of a universal healthcare system is a very important step towards this goal, with the antiviral therapy of chronic hepatitis C providing a great opportunity to demonstrate the benefit of this system. For sure, there is a high number of HCV-infected individuals, and all of them should get the healthcare they need. And this has to come along without the risk of being driven into poverty, because universal access to affordable, which means generic DAAs, and intensified screening to identify those who are infected is warranted. Only then we will be able to reach global control of hepatitis C at the national and international level, which is in line with the declared goal of WHO. In this respect, the topic of this Prince Mahidol Award Conference is a perfect fit, and I want to congratulate the organizers for having put together such an exciting meeting on this important topic. As a basic scientist, I had the privilege to follow the history of hepatitis C from the discovery of the virus in 1989 until the implementation of all oral interferon-free DAA-based therapy. Although it is often perceived that DAAs are equal to the end of HCV, this is certainly not true. Following a quote of Winston Churchill's, one can say DAAs are not the end, they are not even the beginning of the end, but they are perhaps the end of the beginning. Important work lies ahead of us, and increasing universal access to DAAs is certainly one major task. Another one that needs to be considered relates to the question whether we will ever be able to eliminate HCV on a global scale by the sole antiviral therapy. I think at this stage we do not know, and there is no precedence for that for a viral eradication just by antiviral therapy. Therefore, we have to be prepared and should at least consider the development of a vaccine to prevent chronic HCV infection. This is a scientifically challenging task, and given the non-existing interest of pharmaceutical industry, that's a topic that needs to be addressed by scientists working in academia. Without such a vaccine, global eradication of HCV is questionable, and therefore we should get prepared. In conclusion, I would like to thank again the Prince Mahidol Award Foundation for their recognition of my work and for honoring me with this prestigious prize. Thank you.